Yeah, so as I mentioned in previous exchange, so the webinar will be recorded, and then yeah, of course we will broadcast at the same time on the YouTube. But after that, we will also upload to Bilibili in order that for the people who want to
that's
attend.
Okay. All the data is published, so it's you can go online and stuff.
Cool.
No problem.
In that way,
it's live on YouTube now.
Okay.
So after when you arrive to Japan, do you still continue work on the similar subject, or you
no, I will work on now I'm working on plant and bacterial interaction.
Yeah.
And when I go there, I'll work on plant insects and plant parasitic plant interaction. Okay. So like mood system. It's still kind of immunity, but it's not like the same system anymore. And now I kind of work on intracellular receptors, and when I go there, I'll work on extracellular receptors.
Cool. That's nice.
It's like switching a little bit, but not like too far, like from what I know. Yeah.
That's good. How long will you stay at Japan? Do you know?
So the fellowship that I been given is like 2 years, but I don't think I will just stay there for 2 years. I'll probably apply for another one.
Yeah.
I guess maybe like 4 or 5 years at least. It takes like some time to publish work. And then before like I move on to like another area. But yeah.
Good. Maybe after that you can come back to Europe, or I don't know.
Yeah, that's what I think. I think I will come back to Europe to settle down, because I think I quite like Europe. But right now I think it's quite, because I've been in the UK for around 10 years already. I think it's quite nice to go somewhere for like a little bit and then come back later.
Yeah, I think it's better to have the different vision.
Yeah, I think so. It's good to have like a little bit of mobility as well. We have like really bad weather for this like few days. I think that's why the internet has gone off. The whole area in my city, like there's no internet. All the Wi-Fi has been down by this service, and then that's why I was worried. Like, because we didn't have internet for like 2-3 hours.
Yeah, but Pintao told me this morning, so I said
I was really worried because I was like, I thought it was only my house that has the problem. And then I look it up online, and it was like the whole like town.
I saw the news yesterday on the TV. So they are really even in London. They are also plan of the.
I didn't know that. I thought it was just Norwich.
I think it's much more than that. So. This year the weather is quite wild. So.
I'm having a thunderstorm here right now.
Oh.
You might hear like some thunder in a bit, but like it's really, really bad outside right now. Very heavy rain.
Okay. Hope that everything will go well for the weather.
It should be fine. I don't think there will be like an internet problem again. Like, just a cut.
So I think today, since it's more or less a holiday, lots of people went to the holiday. There might be less people to come online, but I think lots of people
that's fine.
We'll check it after that, so don't worry. So.
No problem.
So we can still wait a few minutes.
No problem.
You arrived at UK for your high school already, or
so I came. I came here to do like 2 years of A levels. I did my I did my A levels here basically.
I see.
And then university, and then into a PhD.
Yeah.
Basically.
You did all your education at UK.
No. Like I didn't do like primary like school. My primary and like most of my secondary school was in Macau.
I see. Yeah, that means that higher education. So after
higher education for sure. I did my undergrad in London. I went to Bacterial college for like 3 years, I think. Yeah. So I was in London for a few years as well.
I think 2 years ago, I also have an internship student from the College London. So.
Oh yeah, it's very very nice. London's very nice, I think.
Yeah.
Compared to Norwich. Yeah.
Yeah, we can wait 1 more minute and then we will start.
I don't mind.
Okay, let's start. So, yeah, 大家晚上好，很高兴今天大家能来参加我们 CGM 的欧洲的活动小知道现在欧洲很多地方都慢慢开始去度假去了，还有很高兴还是有朋友继续留守来参加今天的活动，然后那些已经度假的朋友也可以之后会看我们的回播，所以今天的报告也会之后会在哔哩哔哩上有回播，然后我们虽然现在欧洲已经进入假期，但是我们的 CGM 活动呢，欧洲的活动也并不会因此而终止，我们后面一直都会有报告，就还是每个2周一期的报告。然后我们大部分的报告人7、8月份主要是来自英国，看来英国的朋友比较积极，这假期还坚守的工作。比我们的欧洲大陆的朋友好多 ，OK， 然后特别今天我们就有请到我们Quino 的角色来自英国的朋友给我们介绍他的非常漂亮的工作。Quino 他之前在澳门出生，然后很年轻就到了英国，就在英国从 A level 开始读起，然后直到大学，然后再读博士然后博士刚刚毕业，现在继续留在原来实验室做 postdoc， 然后很快就会到日本去做他正式的一个 postdoc 工作。然后today is my pleasure to have Quino to give a very I hope it will be a great seminar on the plant immune system. That I'd really like to invite someone to talk about the plant immune system during our webinar series. So now finally I get a keynote to talk about that nice work just not published long time ago on Nature. So now I give the floor to keynote. So.
Okay. Thank you for a very nice introduction. I'm just going to share my screen. Okay. Yes. So today I'm going to talk about the title of my talk is going to be like mutual potentiation of plant immunity by cell surface and intracellular receptors. So before I go on to my talk, I want to give a brief introduction to myself. So as introduced, I came from Macau. It's like I think most people would know where Macau is. It's in the south of China. It's like a small island that was colonized by the Portuguese for like a while. So China's recently taken it back in 1999. And because it was colonized by Portuguese for such a long time, we have like a lot of quite interesting architecture. We have buildings that it's like these Portuguese like European influence buildings, and we also have like very traditional Chinese buildings and decoration. So it's like quite a nice place. And I think most people in the audience would know that Macau is quite famous for its casinos, and a lot of people came down to gamble every year. I haven't went back for like 2-3 years already, but it's a really nice place and a lot of nice food. So if you haven't been before, I really recommend going to Macau. So yes, I have been in the UK for 10 years. This is my 10th year in the UK. I came from Macau to do my A-levels or high school for like 2 years, and I went to Imperial College London to do my bachelor in biology for around 3 years. And directly I just did a PhD in the Sainsbury Laboratory in Norwich in the lab of Jonathan Jones. And then this work I'm going to present today is the work that I've done during my PhD in JJ's lab. So before I start talking about my work, I want to give a brief introduction of the plant immune system. So like in the natural environment, plants face a lot of different pathogens. It's not like what we grow in the greenhouse. It's all nice and they're not stressed at all. But in the natural environment, it faces a lot of stress. So I want to briefly talk about how the plant immune system works first before I talk about my work. So in the normal unstressed plants, there's defense response is not activated, of course, and there are two classes of immune receptors in plants. So one of them is like on cell surface, which is known as the PRRs, and then you also have immune receptors inside the cell known as NLRs. And this I'm talking about the innate immune system because so far we don't know whether plants have adaptive immune system. But so most of the time when we talk about plant immune system, we are talking about an innate immune system. And so I'm using Pseudomonas syringae, which is like a bacterial pathogen. That's my pathogen model. So when it lands on the plant, so it activates pattern trigger immunity or PTI. So these molecules or patterns from bacteria, or known as PAMs, is recognized by PRRs. So flagellin from Pseudomonas syringae is recognized by the PRFS2 and its co-receptor BAC1. And then once this epitope is being recognized, it activates this kinase domain in the cytosol, and it leads to multiple phosphorylation cascade events. So the cytosolic kinase BAC1 is activated. After it's activated, it activates the NADPH oxidase, RBOHD, that leads to ROS production. And then ROS production leads to callus deposition, which makes the cell of the plants thicker to protect the plants from infection. And MET kinases are also activated following the perception of PAMs. And then the MET kinase cascade activation leads to transcriptional reprogramming. And this transcriptional reprogramming, together with the callus deposition, protects the plants from being infected by different pathogens. And so the bacteria or other pathogens is quite smart. It knows how to inhibit this process. So for using Pseudomonas as an example, it uses a type-free secretion system to deliver effectors, these molecules, proteins, or different types of compounds called effectors, into the plant cell. And then once it's inside the plant cell, it does different types of reactions to inhibit pattern trigger immunity. So it suppresses PTI. So this leads to effector trigger susceptibility. And in turn, plants evolve these intracellular receptors called NLRs, and NLRs basically recognize effectors. So in our lab, we study the effector AVRPS4 that is recognized by the NLR RS1-NRPS4. So intracellular, this is recognized as intracellularly. So Pseudomonas deliver AVRPS4 into the plant, and then the plant NLR RS1-NRPS4 recognizes these effectors. And then this activates the effector trigger immunity, or we call it ETI. And when ETI is activated, it leads to this hypersensitive response, or known as just HR or just cell death. And then you see patches of leaves dying or committing suicide to prevent the pathogen to spread into the other parts of the plant. So this is basically the brief introduction of the plant immune system. And so for a very long time, so we're studying effector trigger immunity, or we're studying how NLR activation leads to immunity. But whenever we say we study ETI, what we do is we usually use bacteria like Pseudomonas, or we use Oomycete like HPA, or we use Ecobacterium to introduce or deliver the effector into the plant cell. So during this process, PRR is always activated. So when we say we study ETI, what we're studying is the NLR activation in combination with the PRR activation. So we're always studying PTI plus ETI. We are very rarely studying ETI alone. So there is like a problem there, which is like the base of my PhD. So how can we study ETI without PTI? How can we activate intracellular receptors without triggering extracellular receptors? So before I joined the lab, my postdoc supervisor Peng Tao, which is an associate professor right now, generated this estradiol-inducible AVR-PS4 line. So in the normal condition, when you don't add estradiol, then there's no activation of immunity. But in these lines, when you add estradiol, you can either spray or you infiltrate into the plant. Then the effector AVR-PS4 is being induced by the chemical. And it's being expressed inside the plant, and then it will directly activate RF-PS4 to trigger ETI without activating PTI. So this is our system that I'm going to be talking about in my work, basically. So this is the system we're using to help us to study ETI in the absence of PTI. Okay. So I divided my PhD talk into 4 parts, and they're all linked. But the first part I'm going to talk about is the potentiation of PTI by ETI. When these lines were generated, we wanted to test whether the activation of ETI leads to bacterial resistance. So what we did is we used this line, we either pretreated estradiol in the wild type, or we pretreated the estradiol in the estradiol-inducible line. So what happens if we add estradiol is that in the estradiol-inducible line, you get the NLR activation. And then after that, 24 hours later, we add the bacteria and then do the infection assay. So on the left-hand side, you have like PTI activation alone in the wild type. And then on the right-hand side, you have PTI plus ETI activation together. And we want to see what happened to the resistance against the bacteria. So it turns out that when we activate the PTI alone, we don't get sufficient resistance. So this is a control compared to PTI activation. As you can see, there's no difference between the bacterial count, or no significant difference at least. But when we activate PTI plus ETI at the same time, then we get like a stronger resistance against the bacteria. As you can see, compared to the control, you get less bacterial count, which means the plant is more resistant against the bacteria. So it seems like the co-activation of PTI plus ETI gives you a stronger resistance compared to PTI alone. So we were hypothesizing like how come when you have the co-activation is stronger than the activation of one just by PTI itself. So we were hypothesizing that perhaps what ETI is doing is potentiating or making PTI stronger, enhancing PTI. That was the hypothesis. So to test this, we did this experiment. Again, we used the estradiol-inducible line. We either treated with MOC or we treated with estradiol. So we added the estradiol into the plants, and we get the NLR being activated, and then we add the PAM to trigger PTI at the same time. And so on the left, again, you have PTI activation, and on the right, you have PTI plus ETI activation. And we're looking at the ROS production during this time. So ROS is like a response that is triggered by PTI. And so it's quite interesting. The result is when we activate PTI alone, of course, ROS burst is like a very standard PTI response. You get this like pretty strong ROS burst. When we activate ETI alone, which is this yellow line here, we don't see anything compared to the MOC, which is the black line underneath it. So when you activate MOC or just ETI by itself, there is nothing. And when you activate PTI plus ETI, you suddenly have like a stronger response compared to PTI. And then it's not really an additive effect because there's nothing in the ETI sample. So what we're seeing here is a potentiation effect. When you have like both activation at the same time, then you suddenly get a much stronger response. And then to add it as a control, we tested this again in the mutant, in the EDS1 mutant, where the activation of RS1 and RPS4 or the NLR does not lead to the downstream responses. So in this line, we don't see significant difference between the potentiation. So it means that it is like NLR dependent potentiation. So we want to repeat this again using a co-activation experiment. So we have 4 different treatments here. We either activate PTI alone, we activate ETI alone, or we activate PTI plus ETI at the same time. And we want to do the ROS experiments again to see what happened over like a longer period of time. And then so we get very similar results. So again, with either MOC or estradiol or ETI treatment, there's no ROS production over 24 hours. And then when you have PTI, then you get this kind of burst for like a 2-phase burst. And then the first phase happens within the first hour, and then the second phase happens between 1 to 6 hours. When you have PTI and ETI activate at the same time, although there's no difference between phase 1 and 2, you see a much stronger ROS accumulation in phase 3 compared to PTI alone. Again, you see when PTI and ETI co-activated, there is a stronger response compared to just PTI or ETI activating by itself. And FLAG22 is only one of the PAMs from bacteria, and we wanted to find out whether this is specific to bacterial response. So we use many different PAMs or like ligands from different pathogen. So we use like a damage response. We use a DAM called PAT1. We also use multiple PAMs from bacteria, use FLAG22, alpha-18, and the fatty acid. We also use some PAMs from fungi and omycete, which is chitin and NLP20, to activate the cell surface receptors. And then just repeat these experiments and trying to see whether we can repeat the same results. So to make the like a lot of experiments short, basically we see very, very, very similar responses. When you add PTI plus ETI together, you get a way stronger response compared to PTI plus ETI or just ETI alone. So the potentiation effect is very consistent, no matter what kind of bacteria or pathogen you're facing, you still get the same response. And then so ROS is not the same thing that we tested, and then we also try to test other PTI responses, whether they get enhanced. So this is step staining. This is looking at hydrogen peroxide accumulation. And when we have MOC treatment, of course, we don't get any stain of the hydrogen peroxide. When we activate PTI or just ETI alone, again, we don't see any hydrogen peroxide accumulation. And only when you activate PTI and ETI together, then you get the strong hydrogen peroxide accumulation. And then I talk about callus deposition a little bit. The production of hydrogen peroxide or ROS leads to callus deposition that protects the plant cell from being infected. And so we did the same experiment, just treat them all for PTI, ETI, or PTI plus ETI. And again, when you co-activate PTI plus ETI, you get a much stronger callus deposition compared to just PTI and ETI alone. And then we also tested gene expression. We tested the defense gene responses, and for multiple PTI responsive genes like this FRK1, NHL10, FOX1, PER4, and then WRKey41. When we activate PTI or ETI or PTI plus ETI for 48 hours, after 24 hours, the activation of these genes is not that strong anymore in the PTI or in the ETI alone. But when you co-activate them together, you get a much stronger genes expression after 24 hours. ICS1 is just as a control here. It's a gene that is very highly upregulated during ETI. So you don't see any difference between ETI and PTI plus ETI. So to summarize part 1, we observed that ETI can enhance PTI-induced ROS production, and this is not specific to bacteria. It's a very broad observation that applies to different pathogens. And then the second part is we also see that ETI can enhance PTI-induced hydrogen peroxide accumulation, callus deposition, and defense gene expression. So all the PTI responses are being potentiated by ETI. And then so the next part is how does PTI enhance ETI? So we want to know more about the mechanism, about how this happens. So to simplify the PTI signaling pathway, when PTI is activated, you get the phosphorylation of the signaling components, like the ones I mentioned before, the big one, and albuhl-HD. And then albuhl-HD activation leads to ROS accumulation and callus deposition that leads to immunity. And MET kinases are also being phosphorylated during PTI, and that leads to transcriptional reprogramming. And together, this leads to immunity. So because we see a stronger response of these physiological changes, as I mentioned before, we're wondering what's happening to the signal as well. So this part, we're basically looking at the signaling components during the activation of PTI and PTI plus ETI. So the experiment setup is very similar. We use bacteria to activate PTI, and we use bacteria plus estradiol to activate PTI and ETI at the same time. And so just focus on this middle graph first. So we activate this and then we collected samples to do Western blot for these different time points, 15 minutes, 2 hours, up to 24 hours. And then we do Western blot to see the activation of these signaling components. So P42 and 44 is basically the phosphorylation of the MEP kinases. And if I only activate PTI, then you get the phosphorylation or activation of MEP kinases up to 2 hours. But when I co-activate PTI and ETI, then the activation gets much longer. It lasted up to 24 hours. So this is very similar for like other signaling components. So albuhl-HT PS39 recognizes the phosphorylation of albuhl-HT. And if I activate PTI alone, you get phosphorylation of albuhl-HT up to 3 hours. But when you co-activate it, you get a much longer phosphorylation up to like 12 hours. And the big one is also phosphorylated, and when it's phosphorylated, it gets like this little band shift. Then you can see this band shift. If I activate PTI, you get this band shift for up to 3 hours. And if I co-activate it, then you can see that the band shift up to like lasted to 24 hours. So the phosphorylation of MEP kinases, albuhl-HT and BAC1 has all been prolonged when you activate PTI and ETI at the same time, compared to just PTI alone. And it's quite important to also mention that when I activate ETI by itself, by just adding estradiol, there's no phosphorylation of the signaling component. I don't see any. So these yellow ones are ETI activation. There's no phosphorylation of MEP kinases. There's also no phosphorylation of albuhl-HT at all. So it's only when you activate both of them at the same time that you get like a stronger activation. This is not an additive effect, like just to mention again. So you might have mentioned from what I've shown earlier that you can see that when I co-activate PTI and ETI at the same time, then you see like a stronger MEP kinase, BAC1 and albuhl-HT accumulation compared to PTI alone. So because we see that, we hypothesize that perhaps we see a stronger activation because the protein accumulates more during PTI plus ETI. So we wanted to test whether ETI leads to stronger accumulation of the PTI signaling component. And then we tested this, so there are many antibodies available to that recognizes PTI signaling components. And when we activate ETI that is triggered by AVR-PS4, then we see very strong accumulation of SOBI1, BAC1, BAC1, albuhl-HT, and MPK3. So these are all PTI signaling components. And during ETI, they are very strongly accumulated. And then we didn't just do this with one effector, we repeat this with multiple different effector lines. And so this is just all different effectors that are being recognized by different NLRs in plants. And you can see like across all these different effectors, you can see strong accumulation of these PTI segment components, at least for protein level. And we want to see whether the protein level increase is due to the transcript level increases as well. So we did qPCR as well. So basically, so this color is correlated to the earlier ones, and then we take the samples at the same time, 4 and 8 hours after activation. And basically, you can see that these genes are very high upregulated after ETI is being activated, and doesn't matter which ETI you're activating, you still get transcript accumulation of all these genes. So both the protein and transcript level are being upregulated. And we want to see like a broader, like more than like just 5 genes. We want to see how many genes exactly are being upregulated during ETI. So we decided to do an RNA-seq on this. Oh, sorry, I forgot about this. So this is just a control. Again, I did the experiment in the EDS1, which is the mutant line. And in the mutant line, you don't see any accumulation of BAC1, albuhl-HT, and MPK3 compared to the wild type. And this is the same for the transcript level. In the transcript level, in the wild type, you see significant upregulation after 4 and 8 hours of the ETI induction. But in the mutant line, we don't see any upregulation. So this is just to prove that it is indeed ETI specific. So as I said, to want to broaden more about how many genes are upregulated exactly during ETI, we did an RNA-seq to want to find out how broad this is. And so we have the wild type estradiol inducible line, we have the untreated samples, we have the estradiol treated for 0 hours, and the estradiol treated for 4 hours. So in this sample, ETI is being upregulated for 4 hours. So this is our test, and these are the controls. We also added a genetic control, the estradiol inducible AVR-PS4 mutant, which will not activate ETI. And then so we also trained for estradiol for 0 hours and 4 hours. And then we have all these 5 samples, and then we did the RNA-seq analysis. And then when we did the parasite score, and then we look at the differentially upregulated genes, we can show that I can see that in the estradiol-inducible AVR-PS line after treated for 4 hours, from clusters 7 and 10, these genes are very high upregulated compared to all the controls. And particularly cluster 7 and 8 is the one that is like very high upregulated compared to the controls. So I took these genes out, there are around 1,000 genes in here. I took the cluster 7 and 8 out and do GO analysis on these genes. So when I did GO analysis, we can see that the top ranked GO terms that has been like recognized are basically the pattern recognition receptor signaling pathway. So the PRR signaling pathway, or PTI, is being very like highly upregulated in the cluster 7 and 8. And then you also get this kinase and transmembrane receptors, which is basically the PRRs and the PRR signaling components that are being upregulated. And then to map all these genes back into the PRR signaling pathway, in this diagram, all these heat maps are showing the PRR or PTI signaling components. So you get the cell surface receptors, you get the MAP kinases, you get the intracellular receptors that are required for PTI signaling. And you can see that the red means it's upregulated, blue means it's not regulated. As you can see, like most of these PTI signaling components indeed are very highly upregulated during ETI. And photosynthesis is basically like a process that is being inhibited during ETI. And this is just our control. As you can see, most of the photosynthetic genes are being inhibited during ETI. But the PTI signaling components indeed is being upregulated. So to summarize, part 2 is we showed that ETI can enhance the activation of multiple PTI signaling components, such as BIG1, RBOHT, and NPK3. And then ETI leads to robust protein and transcript accumulation of PTI signaling components. And this has a genome-wide effect. It's not just a few genes. It's just many genes are being upregulated during these processes. And this is probably have multiple mechanisms that are involved in this. We also tested a little bit of other tests on the effect of protein turnover and translation in our paper, but I don't think I have time to talk about that today. And if you're interested in those, you can read our paper later. So I'll move on to part 3 now. So just to bring it back to more like a biological sense, what does this actually mean? So my supervisor, Jonathan, and another PI, Jeffrey Dangle, proposed this model for plant immunity in 2006. It's called the zigzag model. It's quite a famous model in plant immunity. So I'm going to briefly explain how this works. So when you have PTI activation, when you have the PAMs, then the PAMs come in and then they're being recognized by PRRs, and you get some defense responses, which is the PTI that I was talking about. And pathogens came in, they secrete effectors that suppresses PTI, so you have less defense responses. So bring it back down into this effective threshold of resistance. And this effectors is being recognized by NLRs, which activates ETI, and it gives like a really strong immune responses, like that is stronger than PTI. And then again, the pathogens secrete effectors to stop these processes again, and then so forth. This is why we call it zigzag model, because the plant and the pathogen are fighting to either like defense for the plant or like being successfully infecting the plant. So this is the zigzag model. And in this model, you can see that in the ETI column, what we're showing that is like the ETI activation alone gives you this really strong resistance. But what I was showing right now is that only when you have PTI and ETI activated at the same time, you get this strong resistance. So we're thinking we should reconsider this model and thinking whether this model is actually correct. So I was doing these experiments to show that like perhaps we can slightly modify this model. And so I used infection assay and Western blots to show you how this works actually in planter. When you have PTI, when you have a bacteria landing on the plant, when you activate PTI, and then you get this accumulation of PTI signaling components and activation of MAP kinase. So on the zigzag model, this would mean that when you activate PRRs, you have some amplitude of defense, which leads to PTI activation. You have some defenses. And then when you add the Pseudomonas, that is wild type that secretes effector, then you can see less accumulation of this signaling component and a weaker MET kinase activation, which means that the effector basically essentially suppressing this process. Then you get this effector trigger susceptibility, and this brings the amplitude of defense down to the basal level, or like weaker than before. And then I use wild-type Pseudomonas plus estradiol to activate ETI, and then you get a way stronger response compared to just PTI alone. So what we are trying to propose is that when you have the activation of NLRs, it restores the suppression of inhibition by effectors. And so what we propose that NLR is doing is actually reversing the suppression and to enhance PTI or PRR activation. So this is our new model. And according to this new model, you would need to have both activation of PRRs and NLRs at the same time for you to have very efficient resistance. So that was one of the evidence that I showed, but like a better evidence is that from this model, what's going to happen if we block PTI, then if you block PTI in this model, then it means that your NLR activation is not going to be effective because they work together. And if you block this, then it's not going to work properly. So that's what we're trying to do in the next experiment. So we did like a bacterial growth experiment using either like Pseudomonas carrying an empty vector or Pseudomonas carrying an effector that's being recognized by the plant. So if I use wild-type bacteria that is not recognized by the plant, then it is quite virulent. And you see the plant being quite susceptible. There's quite a lot of bacteria. And if I use a bacteria that delivers APRPS4, which is being recognized by RS1 and RPS4, and the plant is responding to it. So you get this defense response, and you see that's why you don't have that much bacteria accumulating in the plant. This is why you see this difference, which is the activation of PTR plus ETR together. And when I do it in the mutant background of the NLR, so if I mutate RPS4, so RS1 and RPS4 is not functional anymore, and AVRPS4 is not being recognized anymore, and then you don't get any difference. So the plant becomes susceptible again, and then the bacteria can infect the plant properly. So this is the most interesting part. But when I mutate the cell surface receptors, so BAC1 and BKK1 are cell surface co-receptors that are very important components for PTI. When I do it in a PTI deficient mutant, you basically get the same results. So it means that it's not only the NLR that is involved in this resistance. If you have PTI mutant, if your PTI is abolished, you also don't get the resistance as well. So this is a very key result. And then we basically just repeat it with different PRR mutants. So this is FLS2 and EFR. So it's two PRR receptors that recognizes other molecules on plants. And although you don't see a full suppression like the PRR co-receptors, you still see quite strong suppression of this immunity compared to wild type alone. So together, we proved that what ETI is doing is to restore and reverses PTI from the suppression by the effectors. And then we also show that, very importantly, ETI-induced resistance is depending on PTI or cell surface receptors. It's not only the activation of the intracellular receptors alone is sufficient to provide resistance. You need the activation of both cell surface and intracellular receptors together for you to get enough resistance against at least bacterial pathogens. So for the last part, because my talk title was mutual potentiation, and I've only been talking about how ETI can potentiate PTI, but I haven't really talked about the other way around. So initially, when we generated this estradiol-inducible line, and then we added estradiol into the line, and I told you earlier that when NLR is being activated, you can see cell death. We were kind of surprised because when we activate ETI alone, we don't see any cell death. When we treat it with MOC compared to the MOC treatment, to the estradiol treatment, when we activated the NLRs, we don't really see any cell death. And so these are just like control mutant lines, and you can see there's basically no difference in any of the leaves that is being presented here. So we were wondering, how come we don't see HR when NLR is being activated? And because what we saw before, perhaps we don't see any HR is because PTI is not activated in here. So we bring the PTI back. So we either activated the PTI with different bacteria, PAMs, or like just different strains of bacteria. And when you activate PTI alone, you don't get any HR, of course. And when you activate ETI alone, you also don't get any HR. But only when you activate it both at the same time, when you have PTI plus ETI activation at the same time, then your cell death is being recovered and you get very strong hypersensitive or cell death response. You can see that the liver is fully collapsed. And so this is not specific to one specific PAMs from bacteria. We tried different PAMs from different pathogens. We tried bacteria, we tried them from the plant, we try fungi elicitors, or we also try oomycete elicitors. So it doesn't matter which kind of PTI that you inducing, as long as you have PTI and ETI co-activation alone, you get like a stronger HR compared to just ETI by itself. So this is like a universal response. When you have PTI plus ETI, you get a stronger HR compared to just PTI by ETI by itself. And then just to make it even more broader, we try to test this on different effector lines. And again, this observation is very universal. Just by PTI activation or ETI activation alone, you don't see any HR. Only when you co-activate at the same time, then you get a stronger hypersensitive response. And to conclude this part, so this is still kind of preliminary. We still don't really understand the mechanism of that. But we show that PTI can also potentiates ETI by enhancing the HR. And then we currently are trying to work out how come PTI can enhance HR triggered by ETI, but we think that this is something to do with RBUHT and the kinase activation. And this is still under investigation right now. So to end my talk, and I want to give like a proper explanation or like a model of what we think how plant immunity works at the moment. So this is like an animation that I'm going to walk you through about immunity, plant immunity at least. So for I'm going to start with pattern trigger immunity, and when we have the bacteria landing onto the plant and the PAMs are being recognized, and then you get the phosphorylation of the PRRs and you get MAP kinase phosphorylation. You also get NADPH oxidation, you get ROS production, and then of course, you produce this callus deposition and these like downstream responses. And so PAMs leads to PTI, which also leads to resistance. So the bacteria is not successful because you have PTI being activated. And then, as I said, a successful pathogen, they secrete effectors using the typhoid secretion system. So when this is being secreted, these effectors block this process, they inhibit the PTI, and then so your activation is basically gone, and then your PTI is being weakened, and so you don't get any defense, and basically the bacteria can proliferate because there's no defense responses. And what we show in our work is that when effector trigger immunity is activated, you get NLR activation, and you get like a lot more PTI signaling components. But very importantly, even if you get more PTI signaling components, these signaling components are not being phosphorylated or activated. So they're just being lying around waiting to be activated. So this is why ETI alone does not give you any immune responses, because you just get more things without them being activated. And in an actual real infection, of course, you have PTI and ETI activated at the same time. So when pathogen is trying to suppress this process, one of the effectors from the bacteria is being recognized by the NLR. And then so the NLR is being activated, it upregulates all these PR signaling components. And because PTI is still activating, and these two leads to a much stronger activation of these signaling components, and you get a way, way stronger physiological responses like callus deposition and ROS production. And the potentiation of PTI by ETI leads to your very strong resistance again. And then that's how ETI protected the plants by potentiation of PTI. So this is our current model of how we think plant immunity works. And the work that I talked about today has recently been published a few months ago. So our work, it's very nice to have like a lot of people helping me with this work. I think it's a very nice project. Mutual potentiation of plant immunity by cell surface and intracellular receptors has been published back to back by another group, Shifang Qin. We work very closely with Minghan and Shifang from China. And then so we collaborated a lot and exchanged a lot of our ideas during preparation of these papers. And it's really, really nice to see that we can do a back-to-back submission and then have their data supporting ours and have our data supporting theirs as well. So this is 2 back-to-back papers that is presenting very similar stories. And if you want to know more, please go ahead and read of both papers. And then by the end, I just want to thank everyone in JS. I want to thank my supervisory team, Jonathan Jones, my primary supervisor, Cyril Sipfo is my secondary supervisor, Ping Tao Ding is my postdoc supervisor, and he currently is postdoc in JS Lab and helped me with a lot of experiments in this work. I'd like to thank all the TSL and JG group members and all the TSL help team and our collaborators, Minghan, Shufeng, and Qingyang as well. And that's the end of my talk. I'm very happy to answer questions. Thank you.
Yeah, thanks, Pino, for this very interesting and clear presentation. So it's really a very nice work. So,
yeah, thanks.
Yeah, if you have any question, don't hesitate to raise your hand. Or if you don't have a microphone, you can also type your question on the chat. I can ask for you as well.
Yeah. Just stop sharing. Maybe if someone is asking questions, I can just already answer them here.
Maybe I can start with your first question. So yeah, on your last part, you have mentioned that you also need the PTI to enhance the ETI induced HR response, right? So you have mentioned you do not really know the mechanics at the moment. I just wonder, for the anisect data that you show, do you think you might be able to find some hint on which gene might be active, need the PTI to active?
Yeah, so the difficult thing about why we haven't exactly worked out what's going on is that HR is a very ambiguous, unclear process. So it's basically what we know right now, how HR works is that pores are being formed on the cell surface and ions are leaking into the cell. So the cell is dying, but we don't really have any genetic components that is directly linked to HR. We have some, and then we're working on those right now. So I mentioned MAP kinase and RBOHD because these 2 components, when you mutate them, you have less HR and HR is reduced. So we have many of those signaling components. When you mutate them, you get reduced HR. So we think that maybe there's a stronger activation of maybe MAP kinases or RBOHD that is leading to this stronger HR. But that's still ongoing right now. But I think for right now, the key is trying to understand how HR is being triggered in the plants. But I think with just the RNA-seq alone, it wouldn't be enough. I think we will need some more mechanistic studies on how come these are being stronger.
I see. Thanks. It's still a complex process.
Yes.
Any other question? I think some people also work in the plant.
I'd like to ask a question. I'm in Kansas. So I want to ask, so as long as you have both PTI and ETI, you could potentially get an HR. So it doesn't matter which pathogen it comes from. I mean, it doesn't matter which kind of, for example, if the ETI is activated by a virus or something else, and then the PTI is activated by another, different bacterial or something, so it will result in HR. So is that true?
Yeah. So normally, normally speaking, in the actual natural infection, even if you only have one bacteria infecting the plant, to activate, let's say, one single NLR, you usually have many PRRs activated at the same time because the plant has a very big set of PRRs to recognize many different pens and from different pathogens. I'm not sure about whether this will actually occur naturally, but if you activate an NLR that recognizes a bacteria and you activate a PRR that recognizes, let's say, a fungi or like an oomycete or a different pathogen, it still gives you HR. So it doesn't matter which one it's not like it has to coordinate. Yeah, it's just as long as both are activated at the same time, you get HR. This is what we observe, at least. Yeah.
Thank you.
Any other question?
I have one question. Thank you very much for the nice talk. There are also recently published papers that shows the NRRs they can oligolization, so they can form this kind of calcium channels. It seems the NRR pathway is very simple, but in your work, you shows the NRR also depends on the ETI. So I wonder, can you explain a little bit about this crosstalks?
Yeah. So you're asking about the regards of how NRR forms calcium channels and how that plays into our story, I guess. It's a very good question. Thank you. So yes, indeed, there has been some there's been new data recently showing that some NRRs like SAR1 and helper NRRs can oligomerize and form these pores on the plasma membrane or different membranes that leads to HR directly. So how that plays in our story. So we think that with what our results so far, when we activate ETI alone, at least for ours, when in our PS4, we don't see any calcium influx in the cytosol. So it's not every single NRR activation you get calcium influx by pore formation. So I think because PTI also activates calcium channels on the cell surface receptors, I think the activation of PTI and ETI together makes more cytosolic calcium influx. That is why they mutually potentiate each other. But this is still under testing right now. But currently, it's not very clear what role calcium plays in the downstream signaling as well. So we're also trying to work on that at the moment. But for now, I can just say that not all NRRs activation leads to cytosolic calcium influx. Yeah. Does that answer your question?
Yeah. Thank you.
Yeah.
Thank you.
Yeah.
I saw that question in the chat. So
yeah. Okay. So ROS formation in cell. Okay. So for PTI, ROS is accumulated mainly, I think both extracellular and intracellularly actually. But there's been a paper showing that during ETI or during PTI plus ETI, you get a very strong inhibition of the photosynthesis. That's why you have like a lot more chloroplastic ROS accumulating during PTI plus ETI. So I don't think there has been like a direct comparison showing where ROS is formed comparing to PTI plus ETI. And the assay I was using would not be able to tell me because it was like a luminol based assay, and it would just give me signal whenever there's like ROS either inside or outside. But I think during PTI plus ETI there will be more ROS from the chloroplast compared to PTI alone. Yeah.
more question? Yeah, may I have another question? Is that in your model you show that the ETI will increase the compartment of the downstream effect of PTI. So within the stem cells, I just wonder whether this can active also. There are also communication between the other cells or the cell from the other part of the plant.
So that's a really good question. So so we didn't do any protoplast experiment, but Min Han has done the protoplast experiments and showing that when you activate ETI alone, then you get the cell autonomous accumulation of these signaling components, which means that it is, at least we know it is cell autonomous. But the systemic effect, we haven't been able to look at it very stringently because with the treatment on the plant, we can treat the plant. Let's say we treat the plant with a particular area of like the estradiol, but we're not sure whether that estradiol will spread around to the other cell. So currently it's slightly hard to study the systemic effects, but I did some preliminary experiments. We don't see a very strong effect of spreading so far. But I think our system is not the best because estradiol is leaky. It could leak to the other cells. So probably it's better to have like a tissue-specific promoter. That kind of thing would be better to study this. But it's a really good question. I don't know about systemic effect, but we know that it's a cell autonomous effect.
Okay, that will be another scope.
Yeah.
Okay, another question on the chat. So Feng Zhu asked that for the future application to control the pathogen of the crop, based on your results, does apply the effectors to plant to maintain the ETI of the plant is a more efficient method than the induction of the PTI?
Okay, so I think there are two parts into that. So the first part is like the technicality of this. So to induce PTI in crops is relatively easy because you can just spray peptides or PAMs or molecules over the plants and they get recognized. But to activate ETI in the plant alone, you will have to have the effector inside the cell. And these effectors molecules are usually quite big and they won't get inside the cell easily. So I think there's some technical difficulty for that already. And also it won't be good to have like transgenic inducible lines, I'm imagining, like growing as well, because GM it's not like a very welcome thing at the moment. And but for it, let's say if this is not difficult to do, it still depends on the level that you're inducing. Because if you induce, let's say if you induce like a really high level of ETI, then the plants will become very sensitive. Then any small amount of PTI, like microbiome or like any kind of bacteria lying around, would be able to trigger PTI, and then you will get like a death response immediately. So I think it depends on how high you can upregulate it. I think activating either is not a bad idea to protect the plants at all, or to prime the plants from bacterial infection. But we will have to be very careful about how much we're activating because we don't want to kill the plant.
Maybe I agree with you. The OGM might not a good idea. Welcome in Europe, but maybe we can perform some, I don't know, the cost strand to fusion to try to farm a fusion strand that contains some inducible promoter of the ETI.
Yes, I think that's it's doable. Okay. I think what our lab has previously done is we're trying to activate ETI when PTI is activated at the same time by engineering some NLRs that can directly recognize the PAMs. So that was one of the work that was generated from our lab. But it doesn't recognize it very well. But I think if it's doable, I think yes, that would be, if we can have the co-activation at the same time, then we get like a very strong protection for the plant at least. Yeah.
Temple might have a question.
Yeah, you can. Can I ask a question which may not relate it to the story? Yeah, I just saw you made a really good figures and animation to show the process of the pathogen. So I'm just wondering, is there any tools or methods?
Oh, it's quite embarrassing, but it's actually just animation. Yeah, but it's actually just PowerPoint. I made it on PowerPoint animation. So yeah, I mean, it's kind of very, like, I think I have some people asked me before, but yeah, I just coded the element individually, and then like I just moved them on PowerPoint according to the animation panel, like part by part. I can send you those PowerPoint slides, and it will have all the elements inside if you want.
That would be great. Really.
Yeah. It's not difficult once you get the hang of how to move them around. It's not hard at all. Yeah.
Just take time.
I'm also interested. I mean, how can you send it to us? An email or something? How can you share that? I mean, the slides.
In animals, sorry?
I mean, email, or how can you share?
Yeah, I mean, I can email to you afterwards.
Yeah, I can
share those slides. Yeah. I can just email one person, and or maybe I can put my email in the chat box, and then you can email me if you want the slides, and I'll email back. I'm just gonna type it in. That's my email address. And yeah, if anyone wants those slides, could you just email me so I can reply the slides back to you? It's not a big file, it's like a very small file.
Thanks, Bruno. It's very nice to share this experience. That's very beautiful, the animation that you made. So.
Oh, thank you.
I always try to avoid animation because the problem is there are lots of presentation. They all ask the PDF file, so that's the problem.
Yeah, I mean, I also don't know how to use very fancy tools like I don't know, like animators or like Illustrator. So I just stick to PowerPoint. Yeah, it's easiest for me to handle. Yeah.
Any more question on the scientific part or on the technical part on the presentation? Okay. Okay, if no more question, I'd like to send again, Bruno, it's really a beautiful work with a very nice system, well adjusted all the questions. So
thank you very much. It's my pleasure to be here. It's very nice to talk to all of you.
Yeah, and I wish a nice vacation for all of you if you take any vacation, so during the summertime. So hope to see you in 2 weeks. So bye.